Let's look at another important cell organelle, that is the lysosome. Yeah, by definition these are membranous bags with hydrolytic enzymes in them. These are bags. They're also called as suicidal bags. We are familiar with that word, right? So where do these come from? These bud off from the trans phase of Golgi apparatus. We have seen that Golgi apparatus has both cis and trans phase. So from this trans phase of Golgi apparatus, this lysosome buds off. Okay. Now, the lysosomal enzymes are synthesized in rough endoplasmic reticulum and processed by the Golgi apparatus. Okay, the enzymes are synthesized by rough endoplasmic reticulum and are processed by the Golgi apparatus. Now, another important exam bit that you have to remember is the marker for lysosomal enzyme. That's nothing but acid phosphatase. Okay. Acid phosphatase is the lysosomal enzyme marker for lysosome. Okay. Now let's look at the functions of lysosome. The most important one, as the name suggests, is lytic activity. Okay. This is the most important function. That's nothing but destruction of unwanted substances, aged proteins and bacteria. Right. Now, another one is the H plus ATPase channel proteins. They help in increasing the proton concentration, that is the H plus concentration within the lysosome, maintaining an acidic pH in the lysosome, so that it can result in destruction of whatever these we have seen, unwanted substances, aged proteins and bacteria. So, how is the acidic pH maintained? With the help of H plus ATPase channel proteins, which increase the concentration of H plus within the lysosome, resulting in an acidic pH and destruction of unwanted substances. So, these are also called suicidal bags or residual bodies. Can also be asked as a question. So, residual bodies are nothing but lysosomes, right, and they have an acidic pH. Now, why are they called suicidal bags? Because now this acidic pH not only destroys the unwanted substances but also destroys the lysosome itself. That is why they are called suicidal bags. Okay. Now, there are three types of acids that you have to remember in relation to lysosomes. These are acid phosphatase number 1, acid hydrolase number 2, acid lipase number 3. So, all the enzymes starting with acid, you should think about lysosome: acid phosphatase, acid hydrolase and acid lipase. Now, another important concept over here is autophagy. What is autophagy? It's nothing but self-destruction. Okay, now this the discovery of this concept has won a Nobel Prize. So, it aids in survival during starvation. How does this occur? Now, how does self-destruction help in survival? These two words are completely opposite to each other. So, let's try to understand how. What happens in starvation is the lysosome starts to engulf the mitochondria that are present in the cytoplasm. Now, we know that mitochondria has proteins in the inner mitochondrial membrane. Now, when these mitochondria are engulfed and destroyed by these lysosomes, these proteins are released, and energy from these proteins is used for the various functions of the cell. That is the concept of autophagy or self-destruction. That is, they destroy the mitochondria and release the proteins for synthesis of energy.